أنا مبسوطة إن دكتور محمد اتكلم كده عن البايوفيلم، جاب سيرة البايوفيلم في المحاضرة. أنا هتكلم دلوقتي عن الجات biofilm in health and disease. My agenda will include introduction, definition of gut biofilms, role of microbial biofilm in human health, and biofilm formation and human disease, and will end by a summary. My introduction: bacteria appeared about 3.6 billion years ago. Von Leeuwenhoek would first visualize the bacteria in plaques scraped from his own teeth. وده كانت سنة 1683. Bacteria lives as the single free plankton cells or biofilm communities. And biofilm grow through the gastrointestinal tract, which contain millions of microbacteria. Abnormal biofilm result in various gastrointestinal diseases. We have great diversity in human microbiome in our body that can form a biofilm, including many viruses, bacteria, and fungi. So what is the biofilm? That biofilm, by definition, is aggregates of microorganisms embedded in a biopolymer matrix composed of host and microbial compounds and adherent to food particles, mucus, or epithelia. Why bacteria needs to live in a biofilm? Acidic juice and saliva easily to be destroy the free plankton bacteria, and biofilm protected the bacteria from being destroyed. So the bacteria prefer to be grow as a biofilm. This is examples of biofilm in disease and nature. In nature, this is an infected food container. This is a biofilm. They attach it to the surface with fimbria, which causing them to be irreversibly adherent to the surface. In the disease, the most common example is the dental plaque that contain more than 700 species of bacteria and archaea. The biofilm in our dental plaques secrete acidic juice that causes the dental caries. This is the picture of hip prosthesis infected with a biofilm, must be to be removed and replaced. The lifestyle of intestinal microorganism can live in three types or lifestyles, either embedded in a biofilms or freely as planktonic cells, or lives in a biofilm but dispersed from the biofilm to be lives as planktonic cells. So how do biofilm form? There are 5 stages, starting by reversible adhesion. The microorganism adhere to the surface and attach to them, but irreversibly. When the fimbria or when the microorganism have a fimbria or pilia, that causing them to attach to each other by irreversible adhesions, secreting the polysaccharides, causing them to colonize and making colonization. Stage 4 is development. They develop in a form of mushroom-like structure. This mushroom-like structure may contain pores that can allow free flow of water and nutrition. And this is a microscopic picture of one of biofilm containing pores that allow free flow of water and nutrition between this biofilm. And final stage is active dispersals, when the biofilm disperse some of microorganisms that start the stage of colonization, then stage of development, and the cycle repeated. There are many structures that attach to the biofilm and have many advantages in biofilm conformations, starting by capsule, which is thick polysaccharide material that help biofilm in colonization and development. The fimbria allow the biofilms to attach to each other by irreversible means of adhesions. Flagella, according function as swimming and freely mobile the organism. Billa is a thicker form of fimbria and allowing the movement of the biofilm or microorganism on the surface. What's the importance of biofilm? First, the biofilm causing resistance, bacteria to be resistant to phagocytosis. They can collect in nutrition and can help in nutrition exchange between the microorganism. زي ما دكتور محمد برعي قال ان هو البايوفيلم كمان بيريزيست البنتريشن اوف توكسيك موليكيول بكتيريا، بيخلي البكتيريا more resistance to the action of antibiotics. والبايوفيلم has a role as a type of sexual production between bacteria and help in gene exchange as a type of sexual formation. The mucus attached biofilm has a biological factory that can add importance to the biofilm. This biofilm can digest diet, host, and nutrition, xenobiotics, and can form a new metabolic product that have multiple function in health promoting function. This may add as a novel factory for treatment of some diseases. Bacterial lifestyle at the mucosal gut surface under healthy condition, either one of three, as I mentioned before, either biofilm form or dispersed from biofilm to be a free plankton cells or freely living plankton cells. Usually, the organism that tries to live freely is easily to be destroyed. And as I say, living alone is easily to be destroyed. So these bacteria prefer to live in a bioorganism to be more resistant. The hemostatic gut microbiota biofilm throughout the GIT. Over the landscape of our GIT, the microorganism increasing when we reaching to the colon. The mucus layer at mucosal surface changing from gel forming type into more loose form at the small intestine. Human appendix is a special case, as the human appendix harbors dense biofilm compared to other parts of our intestine. Due to its location and narrow side of the appendix, they can serve as a safe house for microorganisms. The appendicular biofilm plus the mucin and IgA formation may help in development of immune system. What about biofilm formation and the human disease? Is the biofilm a real source of digestive dysfunction? And what's clinical importance of gut biofilm? Human infection mostly derived from the biofilms, and altered biofilm are associated with innumerable diseases such as cancer, colon, inflammatory bowel. The field of biofilm research is expected to produce the important tools in the field of patient management. There is actor relationship in biofilm and host interrelation in healthy state. At the type of symbiotic relationship between the health and microorganism, the host side secreting membrane vesicles, mucin, this cause interaction and triggering the immune response between the biofilm and host. The biofilm biography under healthy condition and stable biofilm, they have multiple health promoting function as a type of symbiotic relationship between host and microorganism. When the biofilm is altered, can be reversed into invasive type and can produce several diseases. This is some examples that we have a session for microbiota describing several diseases. Just I mentioned an examples. For biofilm drive colorectal carcinogenesis model. The bacteria dysbiosis alter the membrane permeability, which increase or augment the oncotransformation of the epithelium, and finally resulting in cancer, colorectal cancer. Biofilm also mentioned inflammatory bowel disease and irritable bowel syndrome. This is a very nice paper published by 2021 about the biofilm that discovered to be present in 60% of patients with irritable bowel syndrome and 34% in patients with irritable bowel syndrome. What about the future expectations? The altered biofilm will explain the disease consensus, and many diseases will provide to be caused by altered biofilms. Biofilm may be used as disease markers, and course prediction of diseases will be explained by patterns of biofilm. The response of treatment may also be predicted, and treatment will be personalized according to different biofilms. This is a very nice review article published 2021 about gastrointestinal biofilm in health and disease. And this is another paper or review articles about biofilm with the inflammatory bowel disease, ulcerative colitis mainly, and irritable bowel syndrome. And by conclusion, the biofilms may play a role in health and disease. In different diseases, a biofilm may have a role in disease stratification, marker of disease prognosis, personalization, and treatment. And further expectation will soon be a reality, yet many questions still needed to be answered. Okay, thank you.